So, good afternoon, students.
Good afternoon, sir.
Good afternoon, sir. Good afternoon, sir.
So, students, we will continue with the topic of antibodies. Okay. So, any question which is in your mind, which you want to discuss or ask anything related to antibodies? Okay. So, just raise your hand. Uh, Name the five classes of antibodies. So, who will give the answer? What are the five classes of? Okay, Devanshi.
Sir, IgG, IgM, IgA, IgD, and IgE.
Okay, okay. And okay, so it's already answered. Okay, so let's do one thing. We'll start with quiz. Okay, so I'm just assigning it to the class, and we'll first do this assignment, and then we will continue with the class. Okay. So it contains questions related to antibodies, the initial part that we have done. Okay. Okay, so I have assigned it to the class. So can you all see it?
Yes, sir.
Okay, good. Okay, so now you can start doing it, and maybe in 5 6 minutes we can discuss the answers. So I hope everyone is doing it. So students, start submitting your answers. So students, start submitting. So only half the class has submitted. Students, start submitting. Okay, so I have got replies from Sanjana, Manaswani, Nandini, Janvi, Rupal, Yukta, Sam, Ayushi, Sahiba, Kshitij and Shakti. So rest of the students,
Sir, I also submitted mine.
okay, but still, okay, so now I have 19 responses. Okay, so okay, let me discuss the answers. So the first question was multiple myeloma is, so it is cancer of plasma cells. Okay, so plasma cells are the one which secrete the antibodies. Okay, Benzie Jones proteins are, so they are the excess light chains found in the urine of patients of multiple myeloma. Okay, which light chain is present in maximum population? The correct answer is kappa. Okay, and this is one question in which most of the students have made mistake, right? So only around 50% of the class has attempted this right. So when we are talking about light chain, okay, so only two type of light chains are there, right? Kappa or lambda, right? So some of you have also taken gamma and alpha, so they are not light chain, right? So these two options are gone. Next we have kappa or lambda, right? And in the class I told you that 60% of the population will have the kappa type of light chain. Okay, so the correct answer is kappa. Okay, fourth one is which antibody classes have subclasses? Okay, so the correct answer is IgG and IgA. So if you remember that table which I showed you in the last class, so IgG had four subclasses: IgG1, IgG2, IgG3 and IgG4, and IgA had two subclasses which is IgA1 and IgA2. Okay, and question 5 was you will find pentamer of which antibody? The correct answer is IgM. Okay, so if you have seen, if you remember that table, only IgA and IgM are uh, the ones which can make like a dimer or a trimer, tetramer or a pentamer, but it's only IgM which makes a pentamer. Okay, and when we talk about IgM in detail, we will discuss what is the importance of having a pentamer. Okay. Question 6. Okay. So delta 2, lambda 2. It's the formula of which antibody? Correct answer is IgD. I don't know why 5 students have given wrong answer for this. If you remember, in that table I said D, delta starts with D, so it's IgD. So I think this was the most easy question, right? So always be careful. If it's epsilon, it starts with E, so it's IgE, right? So it's easy to remember this way, right? Okay, so let's start with today's class, and I think we can also have attendance and then we can start. Okay. Okay, so Adish.
Yes, sir.
Adish, you were not visible.
So am I visible now?
Yes. Okay. Atoiba.
Present, sir.
Ayushi.
Present, sir.
Sam. Devanshi.
Present, sir.
Ishita.
Present.
Ishita. Okay, Janvi.
Present, sir.
Kshitij.
Present, sir.
Manaswani.
Present, sir.
Namrata.
Present, sir.
Nandini.
Present, sir.
Nehal.
Present, sir.
Rupal. Sahiba. Sahiba. Sanjana.
Present, sir.
Shakti.
Present, sir.
Shivani.
Present, sir. Present, sir.
Shivani, you were not visible.
Present, sir.
Okay. Tanvi.
Present, sir.
Tridev.
Present, sir.
Vivek. Vivek.
Sir, his Teams is hanging. He messaged on the chat box.
Okay. Yukta
Present, sir.
and Yuvraj.
Present, sir.
Okay. Okay. So let me share the slides and we
Sir,
can.
am I audible?
Yes, Saiba, you are audible, not visible.
Sir, I'm present, sir. Sir, am I visible now?
Yes. Okay. Okay, students, so this was the slide, the last slide in the last class, right? And I'm sure now you have a very good idea of this. So there are different classes of antibodies. So already you know these are 5 types: IgG, IgM, IgA, IgE, and IgD. Okay, so the class is determined by the sequence of the heavy chain, right? So you know the heavy chains. So for G it is gamma, mu for M, alpha for A, epsilon for E, and delta for D. Okay. Subclasses, so as I said, and you also know now, it's only IgG and IgA which have the subclass. Okay. Light chain will always be either kappa or lambda, right? And in multimeric forms, you will only find IgM. So IgM, it's either in a monomeric form or a pentameric form, right? And you may find IgA, it could be a dimer. So mostly you will find it as a monomer or a dimer, okay? And rarely in combination of 3 or 4, okay? So I'm sure it is clear to you, right? So now let me show you this picture. So these are the 5 classes of immunoglobulin, and students, it's a kind of quiz here. Just see this image and try to differentiate. So if you can give me some differences. So see all these uh, classes carefully and tell me if you can find some difference here. So you can raise your hands. Okay, I can see one hand, 2 hands. Okay, so let me just be there with 3. Okay, so so let's start. So all of you give me just one difference. Okay, so Manaswini, what is your point of difference?
Sir, in IgE, the heavy chain has 3 domains, whereas in IgG and IgD, there's only 2. So in the lower part, so IgE has a total of 5. The heavy chains has a total of 5 domains, whereas in IgG and IgD, and it has 4 domains.
Okay, so if we count, so you are saying IgE has 1, 2, 3, 4, 5,
Yeah.
right? 5 domains in heavy
Yes.
chain,
Yes.
and IgG has 1, 2, 3, 4, right?
Yes.
Okay, so this is one difference. So in IgE and IgM, there are 5 domains, right? While in rest other, there are only 4 domains. Okay, so this is one difference. Sam, what is your point? Yukta, can you switch on your camera?
Yes, sir.
And Sam,
Sir, I cannot see the slide right now because your camera is on.
okay, I thought, okay, let me share this slide then. Can you see now?
Yes, sir. Sir, so in IgG and in IgD,
Hmm.
uh, the difference is there is the hinge that connects the upper domain and the lower one. In IgG, there are two. I can see two of them in IgG, and in IgD, there is only one. And also, I wanted to mention in IgA, there is some special type of bonding between the monomers. That's it.
Okay. Okay. So one point is of the hinge region, right? So you can see that there is a difference in hinge region, right? So again, we will come to this point of hinge region. Okay. Yes, Sam, what is your point? Sam, you are not audible. Although your camera is okay. Your I think there is some problem with your audio.
Am I audible now? I just
Yes, you're audible. Yes.
in IgM there's a kink-like region inside
IgM there is?
a kink-like region. If you see in the
Okay, okay. Got your point. Just hold on. Okay, let me share to show you the difference which Sam is talking about. I think Sam is talking about this, right?
yes, sir.
So Sam, this is also there in IgA,
They're in IG and IgM.
right?
Yes, sir.
So this is another difference. Okay. So there are a lot of differences which you will find in these 5 different classes. Okay. And there is another very interesting molecule here, this red one, right? So when we will talk about IgA in detail, we will talk about this uh, molecule as well. Okay. And this is a dimer of IgA. So if two molecules of IgA are held together, they are held together in this way, right? And this is a pentamer of IgM. Okay, pentamer means 5 molecules. So 1, 2, 3, 4, 5 molecules of IgM held together, held together in this formation. Okay, and you can just imagine if it is in this way, it can bind so many different antibody antigens, right? So it can at least bind 10 different, not different, but 10 antigens, right? So that is the importance of having a pentamer. And we will discuss this in detail later, okay? So, length of constant region. So if you just remember what we just saw, and we also discussed about these domains, right? So if you just talk about the length of constant region, so this is almost 330 amino acids in delta, gamma and alpha, right? Where the length of constant region in mu and epsilon is 440 amino acids, right? So in IgM and IgE, there is one extra domain. Okay, and you can see here in this picture, IgE has 1, 2, 3, 4. Okay, so only these 4 are part of the constant region because this one is the variable region, right? So if I ask you the heavy chain, okay, the constant region, so they are 3 in IgG, okay, in IgD and IgA, whereas there are 4 constant region domains in IgM and IgE. Okay, so students, be very careful when you see the question whether they are asking for the constant region domain or the whole of that chain. Okay, are they asking for one chain or are they asking of one antibody molecule or whether they are asking for a pentamer. Okay, so if it's a pentamer, whatever your number is, you have to multiply it with 5, okay, to get the correct answer, right? Okay. Uh, so when these sequences were carried out, okay, so it was observed that for mu and epsilon, it is almost 440 amino acids, whereas for uh, these three types, it's 330 amino acids. Okay, and so these are the major sequences, and these major sequences had some minor differences, right? And on the basis of these minor differences in amino acid sequence, two of the type, which is IgG and IgA, they were further classified into different subclasses, right? So you know IgA has two types, subtypes, IgA1 and IgA2, whereas IgG has four different subtypes, which is IgG1, IgG2, IgG3, IgG4. Now coming to the variable region domain. Okay, so we have talked about the constant region, right? Now we are just talking about the variable region domain, the topmost domain, right, from the amino terminus. Okay, so why it is called variable region? Okay, we need to understand this. So detailed comparison of amino acid sequences of variable light and variable heavy chain revealed hypervariable regions, right? Now हम कह रहे हैं कि जब हमने वेरिएबल रीजन का एनालिसिस किया, ओके, so in that variable region domain, the scientist observed that there are some points which are hypervariable. ठीक है, तो वेरिएबल रीजन के अंदर there are some hypervariable regions, right? So there are three hypervariable regions in mouse and human heavy and light chain, right? So चाहे तुम variable region of heavy chain लो, चाहे variable region of light chain लो, ठीक है, there are three hypervariable region, okay, and these hypervariable region are parallel, right? So अगर हेवी चेन में जिन जिन पॉइंट्स पे है, लाइट चेन पे भी उन्हीं उन्हीं पॉइंट्स पे होगा, ओके, एंड रिमाइंडर ऑफ द वेरिएबल लाइट एंड वेरिएबल हेवी डोमेन एग्जिबिट फार लेस वेरिएशन, ओके, द होल रीजन इज वेरिएबल, right? Some points are hyper variable region, other points they have variation, but it is less variation, okay, and these stretches are called the framework regions, okay. So if we talk about the variable region, so the variable region consists of hypervariable regions and the framework regions, okay. I will make you understand this with the help of a diagram. I hope you have understood this, okay. Hypervariable region forms antigen binding site of antibody, okay. So if this is the V region, okay, in this there is hypervariable region, so there are three hypervariable regions, okay, and these are the 3 points where it will bind to the antibody, okay. So let me make a diagram of it. Okay, so this is your full molecule of antibody, okay. Now you know where are the variable region. So this box is your variable region, right? So from the amino terminus, the uppermost domain, the first 100 or 110 amino acids, they are the variable region, right? So जब इस variable region की sequencing की गई, right? Because we are talking about sequencing ना, so जब इस region की sequencing की गई, now let me enlarge this. Okay, so this is your hypervariable, this is your variable region, right? Now, in this variable region, there are three hypervariable region, right? So, suppose these are the three hypervariable regions. Right? एंड यहाँ पे क्या लिखा है कि इन थ्री हाइपर वेरिएबल रीजन के अलावा जितना बाकी का रीजन है, दैट इज योर फ्रेमवर्क रीजन. ओके, सो बाकी का जितना भी एरिया बचा, दिस इज योर फ्रेमवर्क रीजन. ओके, सो यू कैन जस्ट इमेजिन दिस होल वेरिएबल रीजन डोमेन, right? This domain has few hypervariable regions और जो बाकी का बचा that is framework region, right? एक frame है जिसके अंदर कुछ hypervariable region fit हो रखे हैं. So just think it that way, right? So I think it will become easier for you to understand. And these hypervariable regions are also called as CDRs or complementarity determining regions. Okay, because they are the regions which will bind with the antigen. Okay, so since antigen binding site is complementary to the structure of the epitope. Okay, clear? So students, you have to remember these 2 points. So when we talk about the variable region consists of hypervariable regions and it consists of the framework region, right? So there are 3 hypervariable regions in one domain of the antibody. Okay, so let me explain you this again. So this is the structure of antibody and there are 3 CDRs, right? Clear? And now see this picture here. So this is, so these are the sequences. Okay, so amino acid sequence. Okay, so at first point there is variability. Variability means you will find when sequencing was carried out again and again, you will find suppose 2 or 3 different types of antibodies of amino acid, right? Then at second point, third point, so you can see there is bit of variability, right? But this is very limited, so a difference of around 10. Okay, but at few points, so you can see from 30 to suppose 35, 36, right? So at this point, there is lot of variability of amino acid sequence. Okay, so this point is called as a hypervariable region, right? Similarly, after few sequences, right, there is another point which is hypervariable, right? So there are 3 regions which are hypervariable. They show a large variation in the amino acid which is present there. Okay, so these are your hypervariable region, the red one, and rest of the region is framework region. Framework 1, 2, 3 and 4. Okay, so even for the light chain it is the same, right? Okay, so is it clear? Any question? Any question on uh, variable region domain? Yes,
Sir,
you
why
can.
it is called hyper variable regions as CDR?
Okay, because they are complementarity determining region, right? So if you just understand this term, complementarity determining region, right? So those hypervariable regions, they have a complementary sequence or structure which can help it to bind with the epitope. Okay, so they are complementary, right?
Yes, yes.
That's why it is called the complementary determining region, because these are the points. Now, students, why this variability? Okay, अगर तुम ध्यान से सोचो, जो कांस्टेंट रीजन डोमेन है, इट इज कांस्टेंट, ठीक है, वेरी लिटिल वेरिएबिलिटी. Okay, you take out IgA, do its sequencing, you will only find two types, right? Take IgE, keep doing its sequencing, you will always find the same sequence, right, of the constant region domain. But there is variability in the variable region. So can anyone answer why? Why? What is the need for this variability? Anyone? Just think. Yes, Manaswini?
Sir, so it's an important region and it has to have variability because this is where antigens bind, right? So it has to be able to bind different epitopes of, a particular antigen. So it has to change according to that epitope so that there's proper binding.
Yes, and students, just imagine there are crores of epitopes, right? There are thousands of different type of bacteria, virus, fungi, then allergens, right? So just imagine that these antibodies have to bind to lakhs or maybe more different types of antigen or the epitopes, right? One antigen can have multiple epitopes, right? So they have to deal, these antibodies have to deal with so many different epitopes, right? So अगर ये variability नहीं होती, then there would be need to have thousands or lakhs of antibodies, different types, right? But we have limited types, but because of this variability, they can bind to as many numbers of epitopes as you want, or as many as the immune system wants, right? COVID, SARS-CoV-2 abhi aaya, right? And our system developed antibodies against it, right? कल को कोई और आएगा, our body will again see it, make specific antibodies, right? So difference कहाँ आएगा? Difference आएगा in hyper variable regions में. Okay, clear? Okay, Sam, yes, you had a question.
Sir, do epitopes have any other function other than being a recognition?
Yes. See, what is epitope, right? Epitope is basically a protein, right, on the surface of a pathogen. Now, the protein can have uh, some role, right? So, just for example, हम अभी ACE2 receptors की बात कर रहे थे, right? Or we, in case of influenza, we talk about neuraminidase, right? So, all those proteins have a specific function, right? And antibody makes, so antibodies are made against a specific or multiple epitopes on a particular pathogen, right? So yes, uh, so it all depends, but yes, in most of the cases you will find that that particular epitope have a specific role. Okay, and I have given you two examples here, one of ACE2 receptors in case of SARS-CoV-2, and okay, ACE2 is on humans, right? But
Like
if we, yes, but if we talk about the neuraminidase, it is there on the influenza virus. Okay.
okay. But sir, like if they mutate to like somehow cover their the structural proteins, so wouldn't it be difficult for antibodies to detect them?
Yes. So this is one of a therapeutic approach, right? So what we do is, suppose this is your antigen, okay, and now this is the epitope, okay. Now what I do is, using some other protein or other molecule, okay, I mask this, right? Now if the antibody comes, it won't be able to bind here, okay, because I have covered this. So so I'm not going to discuss that in detail here, but we can develop a lot of drugs, we can develop a lot of different types of antibodies, right? And we can cover this epitope, right? And this, I think, one example is uh, the antiserum, right? So in that, what we are doing is, we are, with the help of antibodies, we are blocking the receptors, right? So the snake venom, it uh, does not binds to its receptor. Okay. Okay, Sam,
Yes,
clear?
yes, sir.
Okay, students, any other question? Okay, so I'll just end the class here because we again have a class now. Okay, so in the next class we will talk about one of our practical. Okay, so if you have any question on this, you can ask right now, or maybe after 5 minutes when we start the next class. Okay, so I'm ending the class now. Okay,
Okay, sir.
and see you in 5 minutes.
Okay, sir.